Hello, thank you for joining us for our webinar series. This month we're doing the series on molecular biology for groundwater scientists. This is our part 3 of that series. Part 1 and part 2 were done by Dr. Frank Loeffler over the last 2 weeks. If you missed those webinars, they are on the webinar section under the resource section of the Microbial Insights website. You can go in and listen to the recordings to catch up. And today we're really honored to have Dr. John Wilson with us. He's going to be completing that third part of the series, and I'm really excited for some of the summaries that he's going to show you and some really new interesting studies that are coming out that he's been involved in. You know, Dr. Wilson needs no introduction. He's a true luminary in the field. Most people have either worked with him or used one of the many documents that he's written for guidance. I know that I use them routinely. Dr. Wilson's a highly respected microbiologist who worked with the Kerr Environmental Research Center in Ada, Oklahoma from 1978 until recently. He led research teams dealing with biological and non-biological processes that looked at how we can destroy contaminants in groundwater. He was a real leader in the EPA in helping to develop and establish a lot of the protocols that we routinely use. And now Dr. Wilson has a consulting company, Scissortail Environmental Solutions. So we're thrilled and honored to have you here with us today, Dr. Wilson, and we thank everyone that's on this webinar. We are recording the webinar today, and again, it will be available on the resource section of our website if you want to go back and be able to review the content of what Dr. Wilson covers. If you have questions for him, please go ahead and enter them into the GoToWebinar software, and we'll answer those at the end of his presentation today. So without any further ado, I'll go ahead and turn it over to you, Dr. Wilson.
Thank you, Dora. This is a privilege for me. When I was a graduate student back in the dark ages, before PCR ages, in 1970 at the University of California, Berkeley, I had a little joke that I shared with my fellow graduate students. You know, I sure wish we could get these creatures to hold up a little sign that says, "Hi, I'm Pseudomonas putida, and I can degrade toluene." Well, today we can actually get them to do that. And so it's my opportunity now to talk to you about techniques and approaches that we can use to use those signs to interpret their behavior and their contribution to environmental restoration. So the goal for this third part of the series is some sort of path forward to interpret that interaction between the presence, the abundance of these organisms, and what they're doing for us. Now there's 3 things that are kind of obvious. We can associate a particular mechanism with an organism, which is valuable and comforting. The second thing is it'd be real nice if we could actually use that to predict a rate constant. I'll be honest with you, we're not there yet. The third thing, which I think really is also a valuable contribution, is we can use those little signs to validate and support rate constants that we get from other directions so that we know that they're actually telling us the truth. I want to talk a little bit about how plumes behave. If you collect enough data at pretty much any plume in 3-dimensional space, and you plot up the logarithm of the maximum concentration at any point in that 3-dimensional space with distance along the flow path, almost any plume will fall into a log-normal relationship. And so you can come up with a rate constant for degradation along that center line of the flow path. Now, most of your wells will fall off the flow path center line, and the concentrations will be lower, so to get this to work, you really do have to have a lot of wells. But almost every plume where I've had this density of data, it falls into this pattern. And other people, even more experienced than I, hydrologists, have seen the same effect. And these are just data from another site. You plot the logarithm, molar concentration of all the chlorinated alkenes against distance along the flow path, and you can bound that upper boundary of the data with a straight line. And that straight line is reflecting a first-order rate constant for degradation with distance along the flow path. So using the little formula there, comparing concentrations with distance along the flow path, you can calculate a rate constant for degradation along the distance. Now that, of course, is very specific to every plume. It's going to be unique to every plume. But we can generalize it and can compare between sites. If we turn the attenuation with distance along the flow path into attenuation with time of travel along the flow path, and so that rate constant with respect to time is simply the rate constant with respect to distance multiplied by the seepage velocity of the contaminant, the groundwater. And so we're going to be talking a great deal about pseudo first order rate constants. And I want to give you go through a little thought exercise with you to give you an intuitive feeling for what different numbers mean. So what I plotted here is what would happen to a hypothetical 10 micrograms per liter concentration at a source of a plume as it moves along the flow path for different values of the first order rate constant. I chose 100 micrograms per liter because that's sort of the upper boundary for many regulators on accepting or at least talking to somebody about using natural attenuation as part of the remedy. So if we have a rate constant of 1 per year, a half-life of 0.69 years, that 100 micrograms per liter can get below the TCE MCL in about 3 years. The rate constant is 0.3 per year. It's going to take 10 years to get to the MCL. If the rate constant is 0.1 per year, it's going to take about 30 years. And if the rate constant is 0.03 per year, it's going to take 100 years. So the ability of these degradation processes to reach our cleanup goals is very sensitive to these rate constants. And so just kind of keep this in your mind over a range of 1 to 0.03. It goes from about the time you can get a regulator to approve a report to the time that a lot of people would like to see it done so they can get it off their books, to a time that's sort of really pushing it as far as how long anybody's willing to wait, to a time that's probably too long to be useful. Now we put all this logic into a decision matrix, which is available on the ESTCP Certif webpage, ER Project 2011-29. And part of that, this was a project that had a very deep contribution from Frank Loeffler. One of Frank's students went through with tutelage from Todd Wiedemeyer and extracted zero first order rate constants for degradation of TCE at a large number of different military bases. And so this is a log of the rate constant that was extracted using a computer model compared to the density of the halococoides cells determined by qPCR. And what a user is invited to do is plot their data on this, and it falls within that blue shape, well, that's offered as evidence that the halococoides can explain the rate constant. Several people have criticized this purely expert-driven approach. So I went back and worked up the data using regression analysis. And so what you see here is the regression line through the data that's within that blue shape, and then 80% and 95% prediction intervals for that data set. And you can see that actually that expert blue shape and the 95% prediction intervals are reasonably congruent. So I'm going to offer this up as probably a better way to look at the data. If it falls between the outer dotted lines, it's a 95% chance it's going to agree with the performance of M&A at the other sites that were involved in this calibrating data set. Now, those dotted lines are actually pretty wide. Back up a little bit. The 80% prediction interval is about a little bit more than an order of magnitude. The 95% prediction interval is getting close to 2 orders of magnitude wide at any value for abundance of D. helicocoides cells. Why might it be that it's so variable? Well, the first and obvious thing is that all the bacteria that have DNA that are picked up by the qPCR analysis may not be active. That probably accounts for much, if not most of it. Dr. Loeffler also mentioned work by Natalie Capiro that shows that for D. helicocoides, the ones that are really active and happy tend to sort to the sediments, and you fail to sample them in a groundwater sample. And for the bacteria that I'm going to talk about later today, we think that many of them might actually be active at interfaces between aerobic water that's got oxygen and anaerobic water that has a substrate. So there are a number of these sort of confounding interactions that add a little, I won't say uncertainty, but a little scatter to the interpretation of a particular measured abundance. If you're really interested in digging into the weeds on this, I recommend this particular article to you. A number of Europeans that have had deep experience in this question got together on a project. And when you went through these sort of things, topic by topic, one of the things that Dr. Meckenstock in particular has pointed out is that the North American concept of redox zones on the left, where the anaerobic plumes tend to go through a sequence of electron acceptors. You've seen this cartoon on the left at many presentations. Well, that's true, but the scaling is wrong. If you actually look at field scale with significant detail and resolution, what you see is the deal on the right. You've got a plume that's pretty much methane producing, methanogenic, and then the transition zones, sulfate reducing, iron reducing, nitrate reducing, oxygen consuming, are very thin, as often as few as a few centimeters. And then it transitions into an aerobic zone. So it all happens, but that transition is rapid and it occupies a relatively small amount of space in the aquifer. What that means is that the bugs that are doing these respirations are accumulating at that interface. And if you put in a monitoring well and you suck water from the whole aquifer, what you do is you blend a lot of clean water with no bugs and a lot of contaminated water with no bugs and a little bit of interface water with lots of bugs, and you get a composite number. And so this could be another thing that might confound some of the interpretation and interactions. As part of that earlier project that Carmen LeBron led with Todd Wiedemeyer and Frank Loeffler, we went through and came up with a decision logic that associated the behavior of a plume with the activity of a particular biological process. And we left out some stuff simply because there just wasn't any real data in the literature on rate constants. We know that aerobic co-metabolism of TCE can happen. It's well documented in laboratory studies, but there was no way to put it in the decision logic because we didn't have any rate constants that we could apply to field scale. Bacteria that degrade a variety of materials using oxygenase enzymes fortuitously, accidentally metabolize TCE to an epoxide. That epoxide is very unstable in water. It spontaneously decomposes to a number of TCE degradation products. So if you have oxygenase activity in groundwater, and that can be supported by either natural organic matter or contaminants such as the alkyl benzenes, or products of anaerobic metabolism such as methane or ethylene that's stripped off of chlorinated solvents, you can get these oxygenases that will fortuitously destroy TCE when oxygen is present. And they're actually really good at it. They can do it when the oxygen concentrations are actually lower than the concentrations that are detected by conventional field methods. So this is a real possibility, and people have been measuring the concentration of these oxygenase enzymes for some time, but they really haven't had a basis of comparison to explain it. So what we're going to do is take a look at the abundance of a number of organisms, or DNA rather, that code for a number of these oxygenase enzymes, and I'm just going to list a few of them. The one that is my particular favorite is methane monooxygenase. Barbara and I were the first people to show that these oxygenases could actually co-oxidize TCE. This enzyme is expensive to an organism. It requires energy in the form of NADP, but it puts one of the oxygen molecules from dioxygen onto methane to make methanol. The other one goes to water. That's why it's called a monooxygenase. There's a phenol monooxygenase that does the same thing. It puts one of the oxygen atoms on the aromatic ring of phenol. There's a toluene monooxygenase that puts one of the oxygen atoms on a hydroxyl group, again on the ring. There's a side chain monooxygenase that puts the oxygen on the methyl group. And then there's a toluene dioxygenase that puts both oxygens on the aromatic ring. I'm going to go back. At the top of the slide, I have some stuff in abbreviation. And that's the tag name for the qPCR primer that's responsible for identifying DNA associated with the synthesis of these enzymes. So there's an MMO primer, a PHE or Phe primer, an RMO primer, a TOL or TOL primer, and a TOD or TOD primer. So we got a new project with the STCP to go back and try to fill in the gaps on the prospects for aerobic co-oxidation of TCE in groundwater at real field sites. And so this is the team that Todd and I put together. The guys in the middle are the ones that did the work. David Friedman at Clemson came up with a I think, very novel and elegant way of actually measuring, assaying for the rate constants for aerobic co-oxidation. Brady Lee at Pacific Northwest National Lab did 2 studies that measured the density or activity of the bacteria. He did qPCR. He also did something called enzyme activity probes. And not listed on the team is Microbial Insights. Microbial Insights had a very substantial in-kind contribution. They analyzed a number of all the water samples for qPCR markers for the oxygenases, as well as for messenger RNA abundance. So this is our approach. Clemson developed that assay. And then Todd and I and Barbara Wilson went out and collected water from 19 wells at 5 sites, sent the samples off to Clemson and to PNNL and to Microbial Insights, where they determined the abundance of cells that metabolized either the enzyme activity probes for the oxygenase enzymes or the abundance of DNA that was amplified. And then again, Microbial Insight looked at the abundance of the mRNA. And then what we're going to talk about in the next few minutes is whether 3, 4, and 5 can explain 2. I want to talk a little bit about why the work at Clemson is so innovative. This is sort of the way people have been doing these carbon-14 assays for the last, actually 50 years. You put a radio label sample of whatever you're interested in into a microcosm or culture media. The bacteria degrade it to produce carbon dioxide. The carbon dioxide leaves the water and is trapped into a sodium hydroxide trap. And as it accumulates label, then that trap was sampled and assayed for the amount of label associated with that CO2. There's 2 problems. The material that has this radio label almost always has something else in it. And so you're looking at CO2 from what you think it is and CO2 from what might be something else. And the other problem is it requires it to go all the way to carbon dioxide to be assayed. So Clemson's done two things that clean that up quite a bit. The first thing is they take their preparation of radioactive TCE, they run it through a column in a GC. They've got a little valve. They know exactly when the TCE peak comes off. And so in that time window, they divert the flow from the GC, not into the detector, but into the serum bottle that is the microcosm of their experimental unit. So they're putting very clean TCE into the microcosm. It doesn't have other stuff that has radio label. The other thing they do has to do with what they detect. TCE is volatile. The degradation products that come out of that epoxide are not. They have oxygen, they're water loving. If you make the solution alkaline, they do not strip if you purge away the TCE. So what Clemson does is they'll take small aliquots from that microcosm over time, make the pH highly basic, blow the TCE out of the water with a stream of nitrogen, and then count the remaining carbon-14 activity using liquid scintillation counting. And so what they're measuring then is the accumulation of that label over time, which is assumed to be the degradation products of the TCE. Now they have some mathematics that accounts for reduction in counts caused by the fact they simply took a water sample. They tease out from that the reduction that's caused by production of transformation products. And so they get a plot of the accumulation of transformation products over time of incubation. And so this is some real data from a site at the Tooele Army Depot in Utah. And the red dots and the red line is the water as received, and they get a rate constant for production of labeled products. That's the 4.2 decays per minute per 3 ml sample per day. And they also get a rate constant associated with the radiolysis of the TCE. This is done in water from the site that's been filter sterilized. It's got no bugs, but we still see the TCE decompose because of the fact that it's exposed to the ionizing radiation. And so what Clemson does is they use statistics to subtract the radiolysis rate constant from the total rate constant to get the biological rate constant. And we're going to compare that family of rate constants now to the abundance of a number of qPCR markers. The ones that are dealing with the monooxygenases it's well described by this article by Brett Baldwin. If you're interested in some of the background, this is the best place to start on that. And so we've got a primer for those 5 different oxygenase enzymes. And they're associated with here in this little table with plausible substrates. Most of them are associated with petroleum hydrocarbons. That methane monooxygenase is associated with methane, obviously. The phenol monooxygenase can be plausibly associated with native organic matter. This is sort of a little cartoon of what native organic matter is like that I pulled off Wikipedia. It's actually for humic acid, which would be a sort of a higher molecular weight. The stuff that's dissolved in the water actually are smaller molecules. But if you look, you see a lot of phenolic structures scattered around the molecule. So there is a very, very good plausible explanation for the existence of oxygenase enzymes that are designed to degrade native organic matter that are associated with the phenolic aromatic groups on native organic matter. All right, now let's talk about the abundance data. One of the things I wanted to include in this project is an approach that's been carried out by a number of people, but probably most successfully at the Pacific Northwest National Laboratory. Brady Lee now leads that group. What they do, a very clever approach, is an organism that's operating an oxygenase enzyme is busy. It's awake. It's actually alive. It has to be to make the NADP cofactors that are necessary for these enzymes to operate. These enzymes are relatively sloppy in their affinity for substrates, and there are a variety of compounds that will fit in the enzyme, operate in the enzyme active site, and produce a product that is fluorescent. So if you take a culture and expose it to this probe, this chemical, after a while the cells will light up under an epifluorescent microscope. So this is sort of the micrographs of what you see under the microscope. The slide in the middle, those glowing dots are bacteria that are now fluorescent under that particular frequency of UV light because they've accumulated the fluorescent product. And so we know that each one of those dots is a cell that is operating an oxygenase enzyme. The frame on the right is the control, so you can see that you really are looking at bacteria. The frame on the left is stained with the particular dye that makes all the bacteria light up. And so PNNL was able to distinguish not only the abundance of cells that had oxygenases and were up and running them, but also the abundance of total bacteria so that we could compare those two populations in terms of their density. So they did these three enzyme activity probes for us. And that just kind of relates the qPCR probe that they roughly map against and the plausible natural substrate for them. Okay, now we're going to show you some data. These are for the RMO gene copy marker. And the guys at PNNL, Brady Lee's lab, did a great overkill. They ran a lot more samples than you would traditionally do so that we could get some idea of the variability. So those error bars are 95% confidence intervals on the means of those samples. And so you can nail these numbers down pretty tight. We also have 95% confidence intervals on the rate constants. And so the Clemson assay, again, is pretty tight. And so these points in the graph are really pretty much points in the graph. So if you look over at the right, what is plotted is the total density of organisms in 8 water samples where we actually got a measurable rate constant. So out of the 19 samples that were assayed, we were actually able to associate a rate constant with 8 of them. And in those 8, the cell density, total cell density varied from about 2 times 10 to the 5th to about 10 to the 6th total bacteria per milliliter. All right, then the red circles are the abundance of cells that were actually carrying this RMO gene. And they are 1/10 or less of that total density. And that's a little bit more heartwarming because that's about what we would expect. It's not going to be the dominant part of the population. And we also see that there's roughly a linear correspondence between increase in abundance, the cells that amplify that DNA, and the rate constants. Now, if you just looked at that, you'd think, well, we've got this nailed, but this is the rest of the story. The little black triangles are the wells where we actually detected the genes, but we didn't get a rate constant. And what I plotted here is sort of this is the masking radiolysis rate constant. We couldn't see the biological rate constant above the rate constant for radiolysis. So we have this particular water that's got lots of gene copies, but we couldn't pick up biology. So what that tells us is when we do get activity, it's only indicative. It's not proof positive that we're going to get the rate constant. It tells us it could happen, but that high abundance is not definitive proof that that rate constant can be expected. We're going to have to find another line of evidence, another way of getting the rate constant to actually nail it down and prove that the site is behaving in that particular way. And so you can't use just the qPCR data to predict and associate a rate constant with the behavior of a particular site. With that caveat in mind, we can put confidence intervals around the regression line through the data for the 8 sites where we did get a rate constant. And here we're comparing the abundance data and rate constants for the aerobic co-oxidation process for TCE to the abundance and rate constants for the anaerobic reductive dechlorination process. This is just for fun. These work by very different mechanisms carried out by very different organisms. Let's see if we can kind of tease out some behaviors. The rate constant of 1 is good enough at most sites to do a lot of benefit. And that's associated with the dihalococoides abundance of about 10 to the 3 per mil. Well, the rate constant of 1 for the aerobic process requires a little bit more than that, maybe 4 or 5 times as many, but it's kind of in the same ballpark. Oops, I'm wrong. I'm sorry, I pointed at the wrong line. Excuse me. Here's 1. And we're way up here at 10 to the 5th. So where it only took 10 to the 3 to get a rate constant of 1 for dihalococoides, it took 10 to the 5 to get a rate constant of 1 for the RMO gene. Again, now that may just be the fact that the RMO bugs are in the water and the dihalococoides bugs are hiding out on the solids, or it could reflect, you know, other cellular interactions. But I wanted to point that out to you, that there are some profound differences. All right, now, if I owned a site, or if I were a contractor working for a site owner and I wanted to interpret data that I got from Microbial Insights and see if there is some hope that this co-oxidation might be useful, I take the abundance data and then compare it first to the regression line. And that's sort of, well, this half of the time it's going to be this faster. In this case, it's about 0.2 per year. And then recognizing the uncertainty. This is the one-tailed 90% prediction line. 90% of the time it's going to be this faster. 90% of the time it's going to be faster than about 0.03 per mil. I go back and I look at computer models of my plume and see if that's enough to do any good. There's another little caveat that I want to offer on this particular interpretation. I told you that sometimes these creatures are associated with fringes, and what the well is doing is mixing the bugs together from regions where they're very highly concentrated with regions where they're very sparse. One way of recognizing whether this is occurring is whether you get water that has both oxygen and iron 3. That's one of the easiest ways to distinguish this kind of behavior. These 3 wells had measurable concentrations of iron 3, so this kind of mixed water possibility is at least possible. So I just wanted to warn you about that. All right, that was RMO. That's looking good. There's another one that looks even better, and that's the phenol qPCR marker. This happens to be data from Microbial Insights. Again, what you see is that the concentration of the DNA marker is an order of magnitude or so lower than the density of total bacteria. We also get a significant amount of can't see 'ems, where the rate constants for the biological process cannot be distinguished statistically from the rate constant for the radiolysis process. So with our caveat previously mentioned, we can put confidence intervals on that regression. And then I would offer this as a way of interpreting the abundance of phi data. In terms of 90% of the time it's going to be greater than this rate, or 50% of the time it's going to be greater than this rate. And then finally, let's take a look at those enzyme activity probes. This particular one is for the CINN, the SEN marker, the SEN probe rather. Again, you see they're measuring that with pretty good precision. The error bars are a little bit wider than they were for the qPCR data, but not too bad. Excuse me. And we have the same issue. We do have a fair number of analyses where we have very high abundance of bacteria that express the marker, that metabolize the probe to produce fluorescent cells, but did not show a rate constant. So we have that same caveat. With that caveat operating, we again can put our regression line through the data and go through that same process to interpret plausible rate constants, not proven rate constants, but plausible rate constants that might be expected to be associated with that particular abundance of cells that metabolize that particular probe. So if you've got a bunch of old, previously acquired enzyme activity probe data from PNNL or otherwise, you now have a mechanism to, at least in a preliminary way, evaluate what that means in terms of rate constants. Now, I told you that Microbial Insights also evaluated the abundance of the messenger RNA in the water samples. And what we saw is, if we saw anything, a much lower abundance. For most of the water samples, for most of the primers, we didn't get anything above their detection limit. In fact, the phenol oxygenase is the one that showed the best response to it. And you can see that it's much lower in terms of abundance. This plots the abundance of the mRNA when we could amplify it against the abundance of the appropriate or the equivalent DNA. And it also puts a regression line through those points. And it's not behaving the same at all. And so it looks very different. And I really don't think in this particular case that the messenger RNA is providing us useful and interpretable information. The enzyme activity probes really go to the same question. Are the bugs there and are they doing something? And so this compares the abundance of cells that react to the CNN enzyme activity probe to the abundance of cells that amplify the Phi DNA marker. And that's a reasonable correspondence. In other words, if you don't have a lot of DNA that's amplified by the PHE or the phenol primer, that much more of that, then you have cells that are actually able to operate an oxygenase enzyme. And then this does the same thing for the RMO qPCR primer. You get a little bit more, maybe, I don't know, half a log more number of hits on the DNA than you do on the cells that actually are open and awake and in business of running an oxygenase. But this tells me the comparison of the enzyme activity probes to the abundance of the qPCR markers, that the qPCR markers are measuring something that's real in the system and not just a bunch of dead DNA or cells that are asleep. So I want to finish up with some warnings against overinterpretation of what I've showed you. We got a fair number of abundance hits in water where we couldn't see a rate constant. So you can't use that abundance number as a sole criteria to associate a rate constant with TC co-oxidation in a plume. You have to have another way of getting the rate constant. Now, the abundance data does have value. One thing it can do is tell you whether it's worth spending the money to get another rate constant with either that carbon-14 assay or assembling enough conventional concentration monitoring data that you can give that to a good hydro modeler and put it in a computer transport and fate model and use the transport and fate model and the monitoring data to back out the rate constant. The other use I think this has, and where I think it'll be applied most commonly and with most effect with regulators, is that you use it as a second line of evidence following the EPA process to validate and evaluate a rate constant that you extracted using the computer models. If you're interested in how that process is done, that's described in exquisite and excruciating detail in the report to that ESTC project, ER 2011-29, that I described at the beginning of the webinar. So there we are, and I would be pleased to spend as much time with you as you wish, taking questions and discussing how this can be applied. Thank you for the opportunity, Dora, to present this.
Oh, thank you very much, Dr. Wilson. That was a great webinar. So again, go ahead and type your questions into the GoToWebinar software. It's such an opportunity to have Dr. Wilson here to answer all of these for us. So our first question, Dr. Wilson says, assuming most biological activity is occurring on the perimeter of a plume, how would you recommend obtaining a groundwater sample to identify the bugs? Do you perhaps isolate the zone in a well screen with Packers? Also, if you do this to collect the sample, should you revert back to standard sampling practices to monitor the plume then over time?
I'm going to ask you to read that again. Let me listen carefully to it.
Sure. Okay, maybe we should break it into parts. The first one says, assuming most biological activity is occurring on the perimeter of a plume, how would you recommend that we obtain a groundwater sample to identify the bugs? Should we isolate the zone in the well with Packers?
First, I think that you're going to have bacteria distributed along the entire length of the plume. You probably actually have more of them close to the source, though maybe not. But I wouldn't accept that most of the activity is at the toe of the plume. But to use the Packers, you have to know the vertical distribution of concentrations and also, ideally, the vertical distribution of soluble electron acceptors or the absence of the electron acceptors. One way to do that would be to isolate zones with Packers and basically just leapfrog your way down the length of the screen. That's obviously one way to go. If the plume is shallow enough and the material is unconsolidated material, I would prefer doing it with a geoprobe and just take a series of vertical geoprobe samples with a lead probe that had a relatively narrow screen interval. You could do it either way. But in terms of interpreting it, I think you're better off, since you're comparing well to well to well along a flow path, to do your analyses on entire well screen, entire well screen, entire well screen along the flow path, and not try to break it up into two-dimensional vertical resolution. What you're looking at with the wells is a diluted impact of a lot of activity in one little area. But what you're trying to describe is actually the composite of the activity across all the material that might be sampled by a well. So in terms of interpretation, though it sounds counter to this caveat that I've warned you about, I'd rather work on wells that penetrated the entire aquifer, so at least were screened across the entire aquifer along a flow path, than to try to look at little vertical sections and compare what would aptly be apples and oranges and pears and grapefruit. All right, second part of that, good question.
Yeah, the second part is, once we collect in that manner, should we revert back to standard sampling practices, or should we continue with the same type of sampling over time?
If I'm going to do sampling over time, I think I'd rather have the whole well sampled, and particularly if you've got a history, a concentration history beforehand. Frank Loeffler gave you this warning yesterday, last week and the week before. Whatever sampling protocol you have when you start, you need to stay with it and not change that process in the middle of a sampling campaign. And so, yeah, I would stay with what you came in with.
I definitely agree with that. We see that, every time you add a variable, it makes it harder to interpret any changes that we see in the data. Okay, the next question that we have, Dr. Wilson, is can you talk some more about anaerobic oxidation?
Anaerobic oxidation of the chlorinated solvents, particularly things like vinyl chloride and cis DCE, was an enthusiasm about 20 years ago. We thought that the iron-reducing bacteria could actually metabolize a lot of these things. It turns out that we can get one culture that can do it for cis DCE, but we can't find it very widely distributed at field scale, and we just don't see it for vinyl chloride. So it's probably other stuff. And I know that's a disappointing thing, but I don't think that anaerobic oxidation is, in my opinion, important. I'm certainly welcome to be corrected by people that have a deeper experience.
Okay. All right, when you think about dihalococoides and some of the things that can inhibit or slow reductive dechlorination, I know Frank covered this a little bit over the last week, so some of the questions that we have talk about, pH is something that we want to think about. Are there other parameters or other co-contaminants that you would be concerned with at sites that might, slow the reductive dechlorination process?
That's something that Dr. Loeffler ought to comment on. Probably the, quote, biggest contaminant that can slow the reductive dechlorination process is nitrate. And the next one is oxygen itself. But if you're in water that's devoid of oxygen and nitrate, then the hydrogen ion, the pH, is the biggest story. I think we tend to not think about things like metals, but because we haven't looked at it, we really don't have a good feeling for it, but that's also a possibility. I suspect that the guy that sent in that question knows a lot more about the answer than I do.
Well, one of the experiences
and if that
we've
person's
had...
willing to come up.
Oh, sorry. I was just going to add to that. One of the experiences we've had is chloroform can be an inhibitor of activity, especially when we're talking about reductive dechlorination of vinyl chloride. So, we've seen some lab studies that have shown as low as 300 micrograms per liter, and then we did see this in the field at some higher chloroform concentration sites, so we felt like that may be inhibitory. I don't know if you have any additional comment on that, John.
Yes, I'm sorry I forgot about that. I looked over your shoulder at some of those sites, and I think that's really very true. However, I don't think that chloroform is a common contaminant at chlorinated alkene sites, but certainly where it's there, it certainly has a potential to have an important impact.
Okay. There's a follow-up about the anaerobic oxidation question. What about anaerobic co-oxidation, like anaerobic methane oxidation?
I don't think anybody's looked at that. If they have, I'd certainly like to know about it. For the longest time, people in the groundwater business thought that methane, once formed in an anaerobic environment, was conservative, was stable. Now, the marine geochemists had known all along that sulfate-reducing organisms and a few other organisms can actually, under complete absence of oxygen, oxidize methane. And when we went back and looked at groundwater systems, the more we look, the more we have seen that occurring. And so anaerobic methane oxidation is a real enough process. I'm just not aware that anybody has evaluated groundwaters where that's occurring to see if a co-metabolism is occurring. That'd be a good area of research. That one needs to be checked. If the guy that sent in the question knows the answer, I'd certainly appreciate a follow-up.
Okay. Okay, so this is a question of petroleum hydrocarbon site. The person has a site where they suspect there's more anaerobic activity than aerobic. So in regards to interpreting the data, once we identify the bacteria, are the methods the same regardless if we're trying to determine rate constants, whether it's aerobic or anaerobic activity?
Well, the methods in both cases are an empirical correlation between a rate constant and an abundance. And I showed you that little slide that compared the RMO genes against the dihalocoicoides genes. And it took 100-fold more abundance of the aerobic gene to get about the same benefit. And so, if you put in that compensating factor in the comparison, you might be able to tease out which is most important at a particular site. But you just can't compare directly the abundance of one to the abundance of the other.
Okay. All right, I think that's all the questions that we have. Thank you again, Dr. Wilson. We appreciate it. It's an honor to have you here on the webinar with us today and to be able to answer all of our questions. I'm sure there'll be other questions that people think about as they start to digest all this information. If you do, feel free to email them to us or to Dr. Wilson, and we'll get answers and get those out to everybody. And again, we'll have the recording available on the website this week, so you'll be able to go on there and listen to this again and send us any additional questions that you have. But thank you so much, Dr. Wilson. It's always a pleasure to hear you speak. I learned so much from you, and look forward to what we're going to do together on this project as we move forward. But thank you, everyone, for your time today. And if you have questions, feel free to contact us, and we'll be happy to answer them. Thank you.